So what we have here, whenever you see it looks like an X shape for a chromosome like that, you know it's been duplicated. It's been copied or replicated. Duplicated, copied, replicated, same thing. This side is the same as this side. And right here is an unduplicated or uncopied. And here we have, if you're looking at this, those are homologous chromosomes, a pair of homologous chromosomes. So can't quite get back there. And for example, this might be chromosome number 1. Both are chromosome number 1. This one would be from your mom, and then this one would be from your dad. So you have one from her and one from him. Chromosome 1. See, chromosome 2, you got one from your mom, one from your dad. So, what do these images have to do with mitosis? Here we have a nice little plant growing. Look at all the life. Look at possibilities right there. Look how dark that soil is, fertile. And here we have an embryo. Look, it doesn't really look like a human right here. It even has little gill slits. But eventually, yeah, it's more like human. Almost like an alien. We got the head pretty big here. Here you have, a cut, like a gash. Ooh, you know, knife went through a little bit. And you're healing up. And here we have a hydra. We got a little bud coming off the side. So what you should think about here, something is growing that requires mitosis. So growth to get bigger from a seed down here. Here we also have growth, this time in an animal, which in this case is a human. Oh, by the way, what's attached here is the placenta. And this little tube that goes with the placenta, umbilical cord. And here you don't have growth. Oh no, here you have repair, because you're fixing what got broken. You need to remake these cells that got broken. And then you don't have growth or repair. You have something new coming from there. And that would be reproduction. And since it's coming off of, it's just a copy of itself, this would be asexual reproduction. But you also need mitosis in sexual reproduction, because in our ovaries and testes, we do have mitosis happening in the early stages of development of the sperm and the egg. So here we have the cell going, the cell cycle diagram here. Most of the time the cell is in interphase, which is all on the outside there. And G1, cell's just doing its normal thing. It's growing, it's metabolizing, you know, burning energy. It's maintaining homeostasis, like what we saw in the potato, when we put the potato cylinders in the different concentrations of water, seeing where the balance of water is. Then you go from G1 to S, and this, you're copying the DNA, just like what it says. Say copy DNA. Then G2, you're going to do more metabolizing, more homeostasis, more growth if necessary, and mainly a prepare for division. Okay, so normal thing, cell in the G phases, and then you have mitosis. And formally, what's happening in mitosis is you're dividing something, and that would be dividing the chromosomes. So we have 40, for humans, we have 46 going to one side, 46 going to the other. And then cytokinesis, this, you've already divided the chromosomes, so now you need to divide the cytoplasm. So all the gooey stuff, you know, after the cell divides, ooh, you got 2 new nuclei, then you need to divide this part. And that's what cytokinesis does. Here we have DNA wrapped up. So here's that X shape we have here, which is, as you guessed it, copied. This side is exactly like this side. And the DNA is wrapped around these little purple things. These are called histones. Like beads on a string. And that's amazing. They know exactly how, or print exactly how wide DNA is. 2 nanometers. 2 nanometers. Remember, a nanometer is a billionth of a meter. So that's pretty small. And look, just don't believe me. They've taken pictures of this, and you can see this structure. So pretty amazing with some crazy techniques. So mitosis, I personally taught it so much. It's like, but we're gonna get through it. Okay, so we got our cell. First thing in prophase, you start to see these chromosomes become visible. Normally they're just a bunch of fuzz. Now they become, you see the structures, and you see these microtubules start growing. Mitotic spindle, okay? These are centrioles right here. And plants don't have these, by the way. Plants don't have these centrioles. So it's a little different with, it's a difference which the animal cells have that plant cells don't. And it's kind of cool because, you know, plants have cell wall, plants have chloroplast, plant cells have plant cells have a large central vacuole, which we don't have, so it's kind of a drag. But yeah, we got centrioles. Moving on, this is like the later part of metaphase. You can see the chromosomes, you know, that are now attached to those spindles, or the microtubules. They're coming out from the mitotic spindle. But then metaphase, you see them lining up in the middle around the equatorial plate, or the metaphase plate, as it says here. And they're getting ready to separate. And remember, they're not pulled, they walk themselves. They basically move towards, they're not pulled. I always thought they moved. I'm sorry, I always thought they got pulled, till shockingly recently, maybe 5, 6, 7 years, I learned they have proteins. They basically walk along this. It's like they're pulling themselves along. Like, get me out of here. Get me over there. And then you see here, this nucleus reforms. The DNA eventually unspool, and they go together. Now, these are the different activities you got to know about. Coiling up, nucleolus, which is, remember, nucleolus. If this is the nucleus, nucleolus is an even darker substance, or looks like the nucleus of the nucleus, basically. And that's where ribosomes are made. Anyway, you got microtubules, no more nuclear membrane. Metaphase, go to the middle, and then they walk to opposite sides, so they separate. They separate, and then telophase, that envelope reforms, and the nucleus reforms, so the envelope is there, of course, and then the chromosomes uncoil and become less visible like that. So, draw some diagrams. Please make them a little nicer than mine. These aren't the best, but, you know, make sure you're drawing these, you know, the nucleus. I got dotted lines, meaning coming apart, chromosomes, nuclear envelope. Spindle fibers forming out of the centrioles. Then you got metaphase. See, they're lined up in the middle. And then anaphase, they're walking to opposite sides. And then telophase, first time, or actually second time, we see these chromosomes by themselves. Now this looks kind of weird. You're like, whoa, chromosomes shouldn't look like this. You're like, no, it shouldn't. But no, yeah, it should. Definitely should. It hasn't been copied. That's the only thing. So really, this is more unusual because this status is not as prevalent as the single status like that. Okay. Oh, so this pretty cool image. We got this cleavage furrow where it's starting to divide. You have spindle fibers pinching along this edge here. This is awesome how this microscopy can show this to us? Cleavage, furrow. Those are R's right there. And so it splits. These are called daughter cells. Don't ask me why not son cells. That's just what they were named. And in plant cells, it's a little different. You'll see basically a new wall becomes formed right here. And boom, vesicles bring the raw materials in there. And start building that. Basically just building until they have that new plate there. So you don't have that pinching. Why? Why don't they just have a cleavage furrow? Well, cell wall. Cell wall is too rigid to do a cleavage furrow like that. So they just got to build a new wall, which is pretty sweet. Oh, there's these pretty sweet images. You see the cleavage furrow here, again, actual images. And the cell wall forming right here. Pretty cool. So we'll see this. I think we'll have time for this lab. It's almost like a regular size cell, but it's just starting to reform, and you see the new nuclei right there. That's pretty cool. Look at that. You got right there the old one. All right, so we've got to talk about cyclones. These are special proteins that control that cell phase diagram. Remember, I'll just kind of draw it again. Remember how we talked about the G1, S, G2, the M phase, and the cytokinesis. So here we go. This is cyclin D. So it triggers that move from in G0 to G1. So to get it started, to come here, you got cyclin D. And then right here you have cyclin, what's this one? E. Cyclin E. And this helps in this stage get that DNA copied up. And here, cyclin A. Activate, also busy in this S phase. Wait a second. So yeah, another DNA replication. Prepares for replication, and then A activates it at the end. And this is cyclin B. Or all this capitals. Doesn't really matter. I don't know why I did that. Anyway, helps to prepare for mitosis, gets that mitotic spindle going. Why did they just go A, B, C, D? There is no C either, so that's kind of weird. Probably because they discovered this one first, and then this one, and then they discovered cyclin C, but it did something else. So then they zeroed D, figured out it did this, and then cyclin E. So we do DNA replication. There's a similar sort of thing going on with that. And then I like to show this. If you walk around California, you'll see a lot of this. It was interesting. Last year it came up. Starbucks had to put a warning because, I forget. There's some anthocyanin. Dang, should have reviewed that before I did this video. Anyway, chemical in coffee in very, very high quantities in lab animals, they find it causes cancer. So because of that, then they have to have this warning that they found a chemical that causes cancer. But remember, these things you have to be in such high concentration. You're not going to get that at Starbucks. On every single car that's sold, you'll see this. So this is on the driver's side window. Driver's side window. And they put this on here because cars are made, there's a lot of chemicals in there to make the plastic, to make the glues. And if you ever get into a car, especially a new car, you'll smell a certain smell. They call it new car smell. So this is basically saying, new car smell could cause cancer. Because in lab animals, they show that chemical can cause cancer. So I just want you to know, you can take, most people don't know you can take the sticker off. Like, it just has to be there. when it gets soaked. I see people driving around. This takes up a lot of space. It blocks out a lot of your view out the window. So I think it's kind of dangerous. Anyway, you see Disneyland here, same thing. And in this parking garage, there's chemicals that are used in the building construction. You know, a lot of adhesives, glues in construction. So every place with a building is going to have those chemicals. So why do we see these stickers? Because we use chemicals in lots of different applications, cars, buildings, and just naturally occurring chemicals. The science has shown to cause cancer at high concentrations. So because of that, people think that we should be warned about it.